Hello, this is Marianne Vertiflor. This is for environmental influences on microbial growth, the salt tolerance testing. First off, microbes exist in fresh and salt water, above and below ground, and on and within other organisms. The diversity of habitats demonstrate the adaptability of microbes. Numerous factors such as temperature, oxygen availability, and pH influence where microbes are found in the environment. So temperature limits the distribution of many organisms. Enzymes, which are catalysts for metabolic reactions, typically function within a narrow temperature range. So oxygen availability also affects microbial distribution. Although oxygen is required for metabolism in many microbes, it is toxic to other species. So let's talk about osmolarity. It refers to the concentration of solutes within a solution. Microbes contain a high number of solutes within their cytoplasm, which makes them more susceptible to osmotic changes. So for high osmolarity, hypertonic, like very salty solution, water from the cytoplasm will move out of the cell. With the loss of water, the stability of the cell membrane and other structures is reduced, and the cell wall will shrink eventually die. On the other hand, if a microorganism is placed in a solution with low osmolarity, hypotonic, such as distilled water, the water will move out of the bacterium. And lastly, we want to talk about halophiles and halotolerant. So let's see. Well, most microorganisms prefer to grow under isotonic or equal environments. So halotolerant microbes are able to survive at high salt concentrations but do not require these conditions for growth. Other microorganisms are considered halophiles because they require a high salt concentration for growth. Alright, so let's get started with the lab testing. Hello, it's Marianne Vertiflor. So today we are doing the testing on environmental influences on microbial growth, salt tolerance, and pH testing. So let's get started. So first, we have the nutrient broth with the 15% normal saline. I'm going to flame it through the lip. There. Now we have the S. cerevisiae. That's the one we're doing it on. I flame this through the lip as well. Okay, so now we're going to use a pipette that was soaking in alcohol. Just going to get 2 drops of the S. cerevisiae. I'm going to place it in the broth. So 1, 2. Just going to flame the lip one more time. I'm just going to flame the lip for the culture as well. Alright, so now we're going to incubate for 48 hours and we'll go check on that. Hello, Marianne Vertiflor. So it's been 48 hours and we're looking at the nutrient broths for the salt tolerance testing. And from what I noticed, it looks like the lower the percentage of this normal NaCl, the more turbidity there is. So let's take a look at this one, the 6 ml. Let's, if you can tell, but there is turbidity there. So that's positive. And I saw negative when it came to the 15%. The CRVCA, it's pretty clear, so I don't see anything there. Same with the Epi, 15%, it's clear as well. But take a look at the 6 ml, 6%. It's a little bit turbid, it's positive. And the 1% for the Epi, there's more turbidity too. Okay. Hello, Marianne Vertify. Here we go. So we are doing the environmental influences of microbial growth salt tolerance testing, and this is going to be on exercise 2. So first we're going to get the nutrient broth. Just going to flame it over the lip. Okay, so next we're going to drop 1 drop of the hydrochloric acid into the 5%. So only 1. Let's see. One drop. And then we have our pipette that's been soaking in alcohol. Actually, so we have the S-CRVCA broth. Just gonna flame it over the lip. We get our pipette that's been soaking in alcohol. We're going to add 2 drops of the CRVCA to the broth. So 1, 2. And the lip again. Just going to cover. Going to flame the lip of the nutrient broth as well. Hello, Marianne Vertify. Here we go. So we are doing the environmental influences of microbial growth salt tolerance testing, and this is going to be on exercise 2. So first we're going to get the nutrient broth. Gonna flame it over the lip. Okay, so next we're gonna drop 1 drop of the hydrochloric acid into the 5%. So only 1. Let's see. One drop. And then we have our pipette that's been soaking in alcohol. Actually, so we have the S-CRVCA broth. Just gonna flame it over the lip. And get our pipette that's been soaking in alcohol. going to add 2 drops of the Cerevisiae to the broth. So 1, 2. And the lip again. Just going to cover. It's going to flame the lip of the nutrient broth as well. Okay, this is for salt tolerance and pH testing. So it's been 24 hours and you can see all of the broths are positive because all of them show turbidity. So for the Epi5, yeah, you can see it's not really clear. I think most of them are all positive. I didn't see any that were clear. Oops. So turbidity, so all of these are positive. This is for the Cerevisiae 5%. So it doesn't matter if it's 5 or 9, they're all positive. Ceri. Yep. Okay, there we go.